Good day to you from Inside Drug Discovery. This video is a brief description of a drug, JOURNAVX, which was approved by the U.S. Food and Drug Administration. This video will mostly emphasize the medicinal chemistry properties of the drug rather than patient outcomes. JOURNAVX was approved on January 30th, 2025. JOURNAVX is approved for the treatment of severe to moderate acute pain in adults. The active pharmaceutical ingredient is suzetrigine. The drug is typically administered orally twice per day as 2 50 milligram tablets in adults. So that is 100 milligrams taken twice per day. The NDA applicant is Vertex Pharmaceuticals of Boston, Massachusetts. The prescribing information insert of JOURNAVX is cited at the bottom of the slide and serves as a primary reference for this video. Let's start with the indication, pain management. Pain management is an unmet medical need. There are certainly available analgesics. They include the opioids, the opioid receptor agonists, the NSAIDs, non-steroidal anti-inflammatory drugs, and acetaminophen. In the case of severe pain, such as pain experienced by patients with advanced tumors and post-operative pain, only the opioids are generally effective. Then again, opioids carry a high risk of addiction, as well as other adverse events. So the safe and effective management of severe pain remains an unmet medical need. Let's see how JOURNAVX possibly fills this need. The target for suzetrigine, the active pharmaceutical ingredient in JOURNAVX, is a voltage-gated sodium channel. There is a family of these sodium channels, and as the name suggests, they allow the flow of sodium ions across a cell membrane. These channels are found in various cell types, including muscle cells as well as neurons. The channel itself consists of multiple proteins, and different combinations of the protein subunits generate the 9 known family members, NAV 1.1 to NAV 1.9. Suzetrigine is a selective inhibitor for NAV 1.8. NAV 1.8 is associated with the pain response, but fortunately does not appear to trigger addiction. So unlike the opioid agonists, NAV 1.8 inhibitors have a mechanism of action that may treat moderate and severe pain without addiction risks. Here are some of the physicochemical properties of suzetrigine. The molecular weight is 473 grams per mole. The log P, this is actually a calculated log P, is low at just 1.21. The number of hydrogen bond donors is 2. Note that many online sites determine hydrogen bond donors based on the number of OH and NH bonds. But the number of donors is actually the number of nitrogen or oxygen atoms that bear a hydrogen. In suzetrigine, there are just 2 atoms, the primary amide and secondary amide nitrogens, that have bonds to hydrogen. The number of hydrogen bond acceptors is 7, same with rotatable bonds. Total polar surface area is 103 square angstroms. All these values show that suzetrigine is well within standard molecular space for oral small molecule drugs. Finally, the target potency is sub 10 nanomolar. All these values are available on the Guide to Pharmacology through the link at the bottom of the slide. Now we have some pharmacokinetic properties. The half-life is fairly long at nearly 24 hours. The volume of distribution is 495 liters, or approximately 7 liters per kilogram, assuming a typical patient is 70 kilograms. Total clearance is 14 liters per hour, or about 3.3 milliliters per minute per kilogram. The oral bioavailability is not reported, and the plasma protein binding is 99%. The only surprising number to me is the half-life. It is quite long at 24 hours. Several days of dosing will be required to achieve a pseudo-steady-state concentration, but a patient experiencing acute pain relief would not want to wait several days to reach full therapeutic levels of the drug. Metabolism of suzetrigine is largely hepatic. specifically through CYP3A4 and 3A5. The main metabolite is M6. Unfortunately, I was unable to find the structure of M6. Since I have to guess, I will say that M6 likely involves demethylation of the phenolic ether, but that is just a guess. Regardless of the structure, M6 also inhibits NaV 1.8 with about a quarter of the potency of the parent drug. M6 has an even longer half-life at 33 hours. Excretion, whether renal or via the feces, includes predominantly metabolites of suzetrigine. Less than 10% of the drug-related compounds observed in the feces were unchanged drug. Based on this observation, although the bioavailability is not reported, oral absorption seems to be quite high. JOURNAVX does carry some warnings and adverse reactions. All the warnings are related to the liver. Patients with severely impaired hepatic function or taking CYP3A inhibitors are at a higher risk of adverse reactions because suzetrigine concentrations will likely be elevated. Suzetrigine is a CYP3A inducer and should be administered with caution to patients taking drugs that are substrates of CYP3A. This is sort of CYP3A inception. Adverse reactions observed in at least 1% of patients taking suzetrigine over 48 hours include pruritus, or itchy skin, muscle spasms, rash, see itchy skin, this could be an allergic response of some sort, and an increase in blood creatine phosphokinase. Importantly, note that symptoms of addiction are not listed as a risk. As has been stated, JOURNAVX is approved for the treatment of acute pain, but there are longer clinical trials underway for other indications that may lead to approval of chronic usage of the drug. That is the story of JOURNAVX, which was approved in a new drug application by the US FDA on January 30th of 2025. JOURNAVX is approved for the treatment of moderate to severe acute pain in adults. Thank you for watching this video. Please consider liking the video, subscribing to the channel, making a comment, or leaving a question. Take care.